if you've got Teams app, you should be able to click on my window where my face is and do something called pin that. It'll have a little push pin icon, and that will make my window that shows me in the room the big window, and that can help you be able to see better. I'm also going to share screen here at the start because I want to show something that's going to be helpful coming up. So in the course resources, I have a link to my YouTube channel. And if you follow this YouTube channel and go to cell biology and click playlist, I have a bunch of videos from the past that are maybe easier to see than the Teams recordings. So I was going to show what videos are in there that relate to these topics. One of them that's going to be useful for everybody, whether you're in person or on Teams, is this one about neuromuscular junction. And when I used to have my channel public, this neuromuscular junction and the glucose transport were my most popular videos to the world because I really worked hard on them to make them nice and practiced and all that. But this one is going to be helpful next week. Also, these two, when we get to muscle contraction, and they kind of go together with the neuromuscular junction video. And then I've also got cytoskeleton, G protein signaling that we're in the middle of right now, cell signaling, muscle contraction, and actin. And some of these may be overlapping. I used to just record any day anybody was going to be absent. There's transport, if you need to go back to that. These are in here by accident. They should be in the plant channel. This one is also miscategorized. But if you're feeling down about this class and you need to pick me up, you can listen to this song. And then I think here at the end is the new worst looking Teams videos. But if you have to miss, or if you want some extra review, go to my cell biology playlist in my channel, and there's some helpful stuff in here. So now that I've showed that, I'll go back and pick up with G protein signaling. Go back and find that slide set. Any questions before we get going this morning? Remember that we will meet in Klein and on Teams at 1:45 this afternoon for your lab session on continuing your papers. And I moved back the deadline to turn in your figure until 1:45 today. So if you haven't submitted your figure that you've chosen, you can do that by 1:45 today. So I'm going to start on the slide that says when the GPCR binds its specific signal. And this is probably going a little bit backward, but I wanted to review what's happening in the beginning. So remember that every G protein signaling network starts with a GPCR. It starts with an extracellular signal, shown in the red, binding to a G protein coupled receptor, and you're going to have 7 transmembrane repeats. And that binding changes the shape of the GPCR on the inside in such a way that it's going to activate a G protein complex, which has alpha, beta, and gamma subunits. And this will dissociate and open up and would be bound to GTP, and as long as the GTP is bound, it is active and it's signaling the next partner in this line. Yeah. And once the G protein complex is activated and separated, one section of it, and sometimes it might be G alpha, and sometimes it might be G beta gamma, but one section of it is going to associate with the next intermediate signaling protein to activate it as long as GTP is bound. As soon as GTP binds G alpha, it has a certain amount of time until it gets hydrolyzed, and that's not very long because GTP is hydrolyzed quickly. But as long as GTP is there, G alpha can associate with this partner signaling protein. And that association causes the partner signaling protein itself to be activated and transmit the signal onward. And that signal transmission will persist until GTP is hydrolyzed and kicks out a phosphate solution. So this would be soluble, and that leaves GDP remaining behind. So G alpha with GDP bound nucleotide will get back together with G beta gamma, and they reform the closed position. And I just want to reiterate here that it depends on the particular cell and the particular signaling network. As to whether the alpha or G beta gamma is the actual signaler. So in some cases, G alpha is going to be the signaler during the active state. In other cases, G beta gamma is going to be the signaler. It just depends on what cell that is and what particular signaling machinery it has.
Could they both be the signal at the same time?
It should be possible for them both to be the signaler at the same time, but I don't know definitely of cases where that happens. But there's not anything particular to prevent that from happening as well. Check something. Nah, it doesn't really. Okay. So, partner molecule. We're going to talk next about what is the partner signaling molecule, and what are the usual ones that participate in G protein signaling pathways. So this is like saying, what are some examples of brown fishhook that could be the next signaler in this signal transduction pathway. So G proteins, when they are activated, will usually be opening or closing an ion channel. That's one option. Active G protein can open or close an ion channel, or it can activate, this one is always activate adenylyl cyclase. Or they activate phospholipase C. Those are the three major targets of activated G proteins. And these two I'm going to go over today. The ion channel thing, we'll see examples of that when we get to muscle contraction. But adenylyl cyclase and phospholipase C are some of the usual targets of an activated G protein. And it's largely going to be one of these at a time. Just one at a time. So we'll look at the adenylyl cyclase. Just skip over the ion channel part. Skip over the slide that says ion channels with cartoons. And go to this one with adenylyl cyclase and the blue squares and dots. So a pretty good percent of the time, an activated G protein, specifically the G alpha, tends to associate with an enzyme called adenylyl cyclase. Adenylyl cyclase. That one is an enzyme, and it is also an amplifier. So notice here we have signal, signal, and then suddenly we have a lot of signal, a lot of diffusible produced signal. This is coming from this enzyme, adenylyl cyclase, that is taking regular, ordinary ATP, same as the cell's energy source, and it is turning it into something called cyclic AMP. I'll scroll this down a little bit so I can reach. So this step is taking ATP, which is abundant. ATP is abundant and, in essence, ordinary, something that is just pretty usual for the cell to have. And adenylyl cyclase is converting ATP into a chemical that is less common, and maybe you could say it's surprising to the cell, or it's really noticeable. It's really noticeable. And this is a signal. Cyclic AMP is acting as a signal. ATP is usually acting as an everyday energy source. So for a cell to have a lot of ATP in the cytoplasm is normal. For a cell to have a lot of cyclic AMP in the cytoplasm is something new and different, and that's a signal, because it's not usually abundant in the cytoplasm. So as long as GTP is bound and G alpha is active, it will turn on adenylyl cyclase. And as long as adenylyl cyclase is on, ATP is quickly converted to cyclic AMP. One more note about this. There does not just have to be one set of these in the cell. Like there may be 50 GPCRs and 50 G proteins, 50 adenylyl cyclases activated. It's not just always one at this one spot in the cell. But we show one as the example for cartoons. So the cyclic AMPs, once they are around, they can diffuse in the cell, and the group of them go and turn on individual intracellular signaling proteins. So each one of these, each one of those can activate a new signaling intermediate. So this is truly an amplification step. This is an amplification. And for each pathway that this works through, keep in mind that we had one, go back to, it started with one signal molecule binding GPCR, activating G protein. G alpha then activates adenylyl cyclase. And now we have a huge number of signaling molecules. So we started with one extracellular red dot signaling molecule out here. It's now turned into a multitude of new signaling molecules. So that's amplification. This is the relationship or the structures of ATP and cyclic AMP. So you see up at the top, that's normal ATP. It's got 3 phosphate groups. And it can be a substrate for adenylyl cyclase, which kicks out a diphosphate into solution, 2 at once. So these are joined, they drift off into the cytoplasm. And as a part of that hydrolysis, there's a chain bond formed between the remaining phosphate and the rest of the molecule, and that's where the cyclic part gets its name. So this is a signal. ATP is normal. The other new thing on this is, yeah, this is a signal, and it drifts around the cell, but we've got to have a way also to turn this off. And the off mechanism for cyclic AMP is a separate enzyme called cyclic AMP phosphodiesterase. So it's breaking that bond. Draw an arrow like snip, that bond, turning cyclic AMP back into regular AMP. And this drifts off in the cytoplasm, and this is also normal, not a signal. It's just part of cell's life. But the cyclic version is the signal.
What does AMP do after that?
AMP is, for the most part, a building block for ATP. So it can kind of cycle back and forth between being produced from cyclic AMP. It can become a building block for ATP, or if the cell needed it, it could also become substrate for nucleic acids too. But it's not an energy source like ATP is. Yeah. It can become nucleotide for DNA, RNA. It can become ATP. So it sits at a crossroads of metabolism, I guess, I would say. So this picture here, I'm going to turn the lights all the way off so that you can see this better. Ooh, lovely. Pops on a rainy day. This is a picture of a neuron. And you can see that it's got cell body here. It's got a lot of dendrites stretching out. And this particular neuron has been exposed to serotonin. And if I had a red marker, I would make the red ball circle that serotonin is the extracellular signal. It's a neurotransmitter, and serotonin binds to a GPCR. So if this neuron is exposed outside to serotonin, 20 seconds later, if there's a dye inside that neuron that responds to cyclic AMP concentration, the whole cell essentially is being flooded with cyclic AMP. within 20 seconds. So what I want to use this for is to have us list in this space in between the pictures, what are the steps of activation? And I'll try to fit it in there. And the reason that we're doing this is that the steps of GPCR signaling activation is something you need to memorize for your next exam. And that takes practice, in my experience, to remember what are all the things that happen. So this is going to become a study guide, essentially, that you can go back and look at the pictures, and here's the list of what has to happen. So we put serotonin into solution, and serotonin is going to bind GPCR. We'll bind the GPCR, and the GPCR will change shape on the inside to activate G protein complex. Try to list these with numbers for those of you following on teams. So number one, serotonin binds GPCR. Number two, the GPCR is going to change shape so that it activates the G protein complex. Number three, G protein complex. This is really more explanation to step two, but G protein complex binds GTP and comes apart into alpha and beta gamma subunits. So number three, G protein complex binds GTP, then comes apart into alpha and beta gamma subunits. Number four, G protein subunit, alpha in this case, activates which enzyme? What's coming next? Brown Bishop.
What is that called, a target protein?
Yeah, it is a target protein. But in this particular model, it's a particular target protein that's going to get activated. What do you think?
Adenylyl cyclase.
Yeah, adenylyl cyclase. And the reason we would know it's adenylyl cyclase is that this is showing cyclic AMP levels. So this neuron is it's like a heat map dye picture. And everywhere where you see the dye in red, yellow, green, that's showing how much cyclic AMP is there. I didn't really explain that very much, but where that picture looks hot colors, those are areas of high cyclic AMP levels. And in this style of signaling, cyclic AMP is coming from adenylyl cyclase. That's how we would know it's adenylyl cyclase. So G protein alpha is activating adenylyl cyclase. Adenylyl cyclase is turning ATP into cyclic AMP. And then cyclic AMP is diffusing into the rest of the cell where it can act as a second messenger. So this neuron on the left, where you see that it's green, those are the first places where serotonin is binding to spots on the cell membrane, on GPCRs. And then from there, if you had watched this in a time lapse, there'll be a little short lag, and then you start to see from all those different spaces, the whole cell start to turn red and yellow, indicating that the cytoplasm is just getting flooded with cyclic AMPs as a signal. But those are the steps. There's two places in this pathway, three really. How could this stop happening or get turned off? There's three off switches.
Cyclic.
Yeah. So one of the off switches, I wish I had a red to do this. So one of the off switches is to have cyclic AMP phosphodiesterase. 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 Remove AMP and just turn it back into normal AMP. So that's off switch number 3. Off switch number 3. Off switch number 1 is for serotonin to stop binding. Serotonin to stop binding outside the cell in the first place. The class of drugs called SSRIs interfere with the normal processes to get serotonin off of a receptor. Yeah. Yeah, there is a second.
I just wanted
Yes.
to
I'm like so 1, 2, 3 style. I should have just started with one. Oh, it's because Tyler said the third one first.
Okay.
No, that's fine. What is the second one? There is a second off switch.
The first one stops the bonding, maybe the second one is stopping adenylyl cyclase.
It does indirectly. How does adenylyl cyclase stop? It does stop. Yeah, it's the G protein alpha not being activated. What causes G protein alpha to not be activated anymore? GTP hydrolysis. Yeah, so the second off switch is GTP hydrolysis. GTP is hydrolyzed into GDP and the phosphate that floats away. So any of those three things could turn off the signaling pathway. This one of the GTP auto-hydrolyzing, that's the most reliable. Like that is automatically going to happen. number 1 and number 2 depend on factors, let's say, but hydrolyzing GTP always happens a little while after GTP binds. So those are the off switches. And if we kept on watching this neuron, it would not stay glowing yellow and red forever. Because if it did, it can't get any new signal from the outside. It would be stuck in an activated.
Hey, Dr. Ong.
Yeah.
I'm sorry, I missed what the first off switch was. Could you repeat that?
Off number 1 or off number 3?
No, off number 1.
Serotonin would stop binding. Something outside the cell causes serotonin to stop binding in the first place. That would be the first off switch. Yeah, so this stuff that we've written down, study this together with the pictures of G protein activation up to this point. Because you'll need to know this process. And I usually, I think, have you draw it or describe it on the exam. And it's up to you, because one time I had somebody just plead with me, please let me just write about it and not draw it. And I said, okay, if that works for you.
You want us to draw the GPCR?
That helps some people. You can describe it or you can draw it, but I'll be looking for, the certain major events.
You were gonna draw, is it like one of
People usually draw it like the book draws it, because that's what they've seen. But I don't know, your book breaks it down into so many steps. You don't have to draw it exactly like the book, but you need to know how these steps work generally and what are the major events, because this early part, steps 1 through 4 that we've got here, well, if we put that in parentheses, all GPCR, G protein pathways do these early events the same way. It's just what is the signal that binds and what's the target that comes next. But that always works the same way. And it's one of the huge, big deal cell signaling networks. Actually, on Professor Rudd's door, there is a poster of G protein signaling networks, and it's got 1,000 protein abbreviations, and like, could be this, could be this. But they all start out this way.
So on the test, are you going to ask specifically which kind, like if it'll be cyclic AMP?
Yeah, you need to know that. So this is one version of G protein signaling. The other 2 versions are the ion channel style or the phospholipase C style. So what I want to talk about next is phospholipase C style, because it starts out the same until it gets to this step right here. So I think what I'll do is leave this up. I'm going to leave that up and hide it. And kind of talk through how is the phospholipase C kind of pathway different than adenylyl cyclase kind of pathway. We won't write anything about this slide that starts with signal transduction components. But what this is showing is sort of a simulation of, those are the early steps. We get to the point of activating adenylyl cyclase to produce cyclic AMP. And this is saying that one thing that cyclic AMP can do is to activate this other protein called protein kinase A, which then goes into the nucleus and regulates transcription factors. This stuff, I do not need you to know details of from this exam. It's just an example of once cyclic AMP is made, it can act on further targets, and this particular style would lead to new transcription, new translation, and a slow response. This would be a slow response. This is not the only thing that cyclic AMP can do, but it is an example of one outcome that some cells would use to respond, in this case to adrenaline, was the original signal. Okay, now we have a new style, a second style of G protein signaling. And I know there's a lot to look at on this slide. So first, notice the early steps are the same in concept. There's an extracellular signal that binds to the GPCR that activates G protein complex. G alpha is bound to GTP. It comes apart from beta gamma. What's new, and that you can circle on here, is we're activating a new kind of enzyme called phospholipase C. So it's this molecule on this slide that's the most key thing, phospholipase C. And phospholipase C is being activated by the beta gamma, G beta gamma. Notice that phospholipase C is up here at the membrane, so it happens to be nearby. It's already up at the membrane. It's kind of near to G beta gammas, waiting to be activated. And what it does when it is activated, we get a hint from its name. So it's called phospholipase, meaning that it breaks apart phospholipids. And it was the third one discovered. That's how it got the C name. It turned out to be the most important. But what it does is it will take a particular type of membrane lipid called an inositol. This sugar here is an inositol sugar. And it's linked to a phospholipid, and these are particularly in the inner leaflet. So we've got a phospholipid here shown in the red, and it's linked to this inositol sugar that's got 3 phosphates on it. And when phospholipase C is activated, it cuts right there between the red and the yellow circle. It cuts that off, leaving 2 pieces behind. A diacylglycerol on its own, which can then, it can drift laterally in the membrane. And the other piece that comes off is inositol triphosphate, which is abbreviated IP3, inositol triphosphate. And inositol triphosphate is able to go and act as a ligand. So they're saying that now pretend that this is red dot ligand. And it goes and opens a ligand-regulated ion channel in the ER, which lets calcium come out into the cytoplasm. This part over here, you can draw a circle around that and kind of draw a line through or put it in parentheses. That's a little bit too much. That's a little bit too many steps to have you learn, I feel like. But getting to the point of having inositol triphosphate drift over to ligand-gated calcium channels in the ER let calcium come out. This is another diffusible signal. Calcium in the cytoplasm is like, oh my gosh, there's so much calcium in the cytoplasm. That's weird. That's a signal. This is another way that G protein signaling can take one extracellular signal molecule and turn it into a big, splashy calcium wave. A cytosolic all through the cell signal is to let calcium out where it's been packed into the ER. This style is considered to be the phospholipase C pathway or the IP3 pathway. I'll be calling it phospholipase C pathway.
Does the phospholipase C pathway have to do anything with muscle?
This is not how muscle gets activated.
Right.
Muscle does get activated by a calcium wave coming out into the cytoplasm. But that calcium wave for muscle has a different way of releasing the calcium wave. And cells that use, like muscle cells wouldn't want to use this pathway as signaling because they're getting calcium waves all the time just from contracting. But other cells in the body that aren't muscle would use calcium wave not to contract, but as like a signal to do something besides contract. But the drama of having that much calcium in the cytoplasm would cause an equally big change in the cell if it's not muscle. Get it to do something important. Yeah. And then the mechanisms to get calcium back in would be similar. But that's a good question. It is a calcium wave, and muscle cells use calcium waves, but this is not how they start. It's a whole separate.
Yeah, I know the
Yeah.
I know the pathway from us. I just wondered if they said anything to contribute to it.
They both result in a calcium wave, but this has a whole different activation, and it's for a different purpose. But having calcium waves to cause something dramatic to happen is pretty ancient in cells history. Muscle just became like a specialization of it, I guess.
What is the purpose of this pathway?
It has a lot of different purposes. So getting calcium wave in a non-muscle cell, I think that's coming up next, maybe. Maybe I need that picture. This is the same picture, it's just reminding that if you open gates to the ER, tons of calcium is going to come out, and that's dramatic. Let me go ahead to this picture. I'll go back to the previous. I'm on the picture that says cytoplasmic calcium can also bind to and activate. Cytoplasmic calcium is so useful and widespread as a signal that extra calcium activates this whole class of separate proteins called calmodulins. And this is a picture of a calmodulin, and it normally has this kind of configuration, like so. But when it binds calciums, it changes shape and it does a weird wraparound shape change that then lets it cleave another protein. So just having a lot of calcium in the cytoplasm causes activation of a whole class of protein calmodulins that then go and do other stuff. And the other stuff that they do is quite diverse. But they're generally going to cleave proteins or go and phosphorylate them, and they can do all different kinds of things. So the outcome of calcium signaling, it depends on the cell type, and it's pretty diverse. You had a question too? I just need to say yay! You know what I'm really good at? Answering a question that the person didn't ask. Like even with my children. Like, Mom, that's not what I want to know. Because I misunderstood their question. So here's a similar style of picture to the neuron that we saw before, but this one is an egg. This is an egg cell, or oocyte. And the arrow up here is depicting the sperm that won the race and first penetrated the egg cell. And that ends up resulting in a GPCR signaling pathway that starts from there, and it's using phospho pathway. So the egg cell now has a calcium responsive dye. This is a fake situation. It's got a calcium responsive dye put into it. And pretty quickly after the sperm fuses with the egg, or if you artificially trick the egg into thinking that happened, you start to see calcium accumulate from there, starts spreading all over the whole cell. This becomes an amplifying signal. And by 40 seconds, half the cell is completely flooded with calcium. Similar process, and it has a similar start, but let's list what are the steps in this that are different than adenylyl cyclase pathway. Think how I'm going to do this. You all probably have, however you want to, here's how I'm going to do this. I'm going to erase from the point that it changes and then write some new things. So for what we've written so far, the first thing that changes out of steps 1, 2, 3, 4 is when we get to step 4 and it says G protein alpha, we can erase from alpha onward. And we will have different steps after that point. But everything that came before is going to be the same. Same stuff. We have binding of an extracellular signal. We still have a shape change of the G protein complex. That shape change causes GTP binding. That leads the alpha and the beta gamma subunits to come apart. But in phospholipase C signaling, G protein beta gamma binds and activates phospholipase C. Binds and activates phospholipase C. Then we have phospholipase C cleaves, cuts, hydrolyzes. A certain kind of membrane phospholipid. And it will turn that phospholipid into IP3 and the diacylglycerol. And then basically the last step is that IP3 binds and opens calcium channels. Calcium channels where? In the ER. I'm going to spell that out, endoplasmic reticulum. So in this style, the outcome is a calcium wave. And then the calcium wave can cause the cell to do other things, but the big diffusible signal through the cell is calcium wave. Mhm. Let me go back and show that, because that's an important question. So the question is about what's cytosolic and what's endoplasmic. So this particular style that uses phospholipase C, there's two membranes that we have to be thinking about. The first one is the cell membrane, or regular plasma membrane here. The signal is coming from the starting signal that starts all of it is coming from outside the cell. So this up here is representing outside the cell. And then everything between plasma membrane and endoplasmic reticulum. So this gray down here would represent endoplasmic reticulum membrane. Everything in between that is happening in the cytosol. So we've got certain proteins that are kind of linked up here at the membrane. The GPCR is in the membrane. The G protein is membrane linked. The phospholipase C is membrane associated. The inositol phospholipid is membrane associated, linked, I guess you would say. And these are all together, but when you cut off an IP3, it can float, and it does, and it will diffuse over to the ER, and that's like a way longer distance than they're showing here, but they got to get it on the same slide. Inositol triphosphate can diffuse or float to where the endoplasmic reticulum is, and there are ion channels there, which are ligand gated. And when inositol triphosphate binds those, they come open more often. They open up more often. Calcium's going to come out because its gradient is oriented this way. Yeah, does that answer? That's one of the things I said. Does that answer what you're asking? Because I know I tend to go on about something that you're not asking. That's a good question. I want to stay on this picture for just a second. Once a calcium wave has been released, that also has to be turned back off. That also has to be turned back off. So remember that there are calcium pumps. We've got calcium pumps in the ER that as soon as calcium comes out, they're going to spend ATP to push it back into the lumen. So that's going to be one of our off switches for this this style of signaling. Okay, I guess I could go ahead and add that onto our list. Okay, so this style, remember, this is the phospholipase C style of G protein signaling. The early steps are the same steps as an adenylyl cyclase pathway until you get to the opening or the activating of the G protein complex. Beta gamma activates phospholipase C, and then we have these other things happening. One of the important off switches for this pathway, the off switch number 3 for this pathway is that we have calcium pumps are going to move calcium back into the ER. It's kind of like if you open a gate to a cow lot, not a cow pasture, but a cow lot, where they're all packed in, the cows will start running out, you know, as soon as they realize what's happening, and they'll start running out. And people will immediately start trying to get them back in, but the cows are going out faster than the people put them in. So that's the off switch for calcium wave for this pathway. This is definitely a good time to take a break. And let me also say, I think this is the toughest lecture of the whole semester as far as all these molecules and all this causes this. And so we might have slightly longer break than usual. Also, I have no idea what time it is. I didn't remember my watch or a mask this morning. It's so bad. I got an umbrella. Got that. We'll have a break right now. Oh, it's not too bad. It's 9 o'clock. I also have to go to the bathroom. It would be useful if I put up a blank slide and took a picture of that. People that can't see it well. Break. I'll do that after I go to the bathroom.
I've got red shorts. I've got like a white bodysuit that I never wear, so I'm gonna paint polka dots all over it.
That'll be easy.
I can paint, yes. And then I can easily make that little money. That we all think we're But my teammates and I went to the party scene yesterday and I was like,
They're so expensive.
I know.
Hey, Kate.
Oh. I just get to do.
Sorry.
That's
I forgot about you. Yes, Josie.
Are you still doing the lab tonight? Is there a lab for comparative tonight?
I haven't
I think
seen.
we have 3 weeks to do
Yeah,
it.
he said we have a lot of weeks. Are you all hiding or where are you?
No, I don't get back until next Friday.
Okay. Well, where's Emily? Where are you? Emily.
Having the quarantine.
No.
Yeah, so next week you may have to start the cat dissection by yourself.
Yay.
I can. Yeah. I probably also could have somebody go with me to attempt to start it today.
I mean, you don't have to start it today. That's okay.
Okay. I mean, I'll start it next week and I'll have also somebody with me too.
Okay, but I was gonna ask when you start, can you text me and then call me on Teams because I want to be there in spirit.
Yes.
I appreciate it.
Do that, and I'll do the dissection, the first part. Oh wait, no, I'm really okay. The only thing that I'm really bad at is uploading stuff.
Yeah. Yeah. Just don't mutilate our cat, please. Like that's all I ask.
Hey, if there are tears involved while I'm doing this, don't. Just tell me the cat was bad or something. It's gonna be really hard for me. Oh my gosh, look at this little boy. He's a little painter. Look how cute.
I like it when people dress their little kids up as little
Yeah.
old people.
Yes.
It's so cute.
Like, this little boy's Colonel Sanders. Does anyone have a child around here? So, what is this? This is so scary looking. Oh my god. Well, we're Candy, God bless. Someone dressed their child up as a homeless person. That's terrible. Oh my.
Costumes.
This one's a cupcake. Yep. A pineapple.
Those would be easy. I think any type of food would be.
Ooh, yes. I was just gonna dress up in a garbage bag and call myself garbage.
For Halloween.
Yeah, but I'm excited to be a gumball machine.
Tomorrow, right? If we were gonna dress up.
Yes.
That's one of the things I love the most about King usually is that we have this great tradition of faculty dressing up for Halloween.
Yes.
But I'm not going to this year. Just can't even. I need to get that again because this is totally relevant. Yeah, it's good. Okay, switch slide sets here to enzyme coupled receptors. It probably seems like I'm way behind, but not so much. It's a short. Oh, it is short. It is short, but it has a lot of details.
So we're not going over ion channels.
No, because when we get to, like, we kind of saw an example just now of how ion channels work, and we're going to see it again in muscle. And that particular one, I know that whenever people have had anatomy and physiology 1 and 2, or mammalian physiology, they will have seen a lot about ion channel activation. So, and if not, like, I make sure that I talk about it enough in this class that this is the only class you're ever going to talk about ion channels, that you have a decent understanding of how it works. I made it through my undergraduate without actually knowing a lot about ion channels, and I did okay. Because in plants, plants don't have muscles or neurons, and they do use ion channels in some cases, but it's not as common as with animals. So we're now going to start looking at a separate kind of signaling altogether. Ion channels are one of the major types of signaling. G protein pathways are the second, and this is the third. And this style is called enzyme coupled receptors. And I think there was, actually, now that I think about it, there was a particular slide in the other slide set that I was supposed to go back and introduce this. So let's go back and find that, because I said I'm going to skip over and then come back to this when we get to the appropriate section. Yeah, here we go. Okay, so ion channel coupled, we've kind of seen a little bit about that already with the calcium channels. And then we've got the G protein style. And then we have this enzyme coupled style. So since I had skipped this previously, I want to use this to kind of set the stage. Enzyme coupled receptors in animal cells are important, but there are not as many of them as G protein signaling pathways. So animal cells tend to use G protein signaling pathways a lot, and ion channels pretty commonly. And the enzyme coupled receptors are important, but they are not one of the, they're the third in popularity, or third in how common they are for animals. But they start also with an extracellular molecule, but the extracellular signal is a dimer. So we're kind of starting off with a subset of, if the extracellular signal is either a dimer or a signal that can cause two receptors to come together. This is a different style. See how you've got to have the signal, and then you have to have two other receptors that the signal is holding or binding to at the same time. Are you missing this slide?
I guess so.
It's like way back in the middle of the intro to protein signaling and G protein receptor. And it's, if you don't have it or can't find it right now, it's not that big a deal. But the key part is this style of signal transduction requires a triple bind at the start. At least a triple bind. So the signal is cooperatively binding to two individual receptor proteins and making them close together. So the signal can be a dimer that brings two identical receptor proteins together, or the signal can be a binder that half of it binds one kind of receptor and half the other, but they have to be all together with at least three parts to initiate the signal. So this whole pathway works by cooperative. The start of it works by cooperative binding to bring two membrane receptor proteins into close proximity. So maybe you could think about the extracellular signal in this case as like a peacemaker, or the kind of person that's like, let's all sit down together and work this out. Let's get the people at the same table and work this out. So the proximity of the receptors is what activates the receptors. The proximity is the key. When you get those receptors close to each other, that's their activation. They got to get close to each other. So we're going to look most at this style right here. But see how when these receptors were further away and not close together, they were not active. But when the signal binding gets them close together, they activate each other. They activate each other, or they activate each other. Sometimes in the past, I have used the concept of people being codependent, that they activate each other, but I don't think that's quite right. Codependent is kind of a pathology more than a benefit. Okay, so now we'll go back to the usual actual slides about the most well-studied example of an enzyme-coupled pathway. We're going to look at one particular example of enzyme-coupled pathway that is the best studied, and it's the receptor tyrosine kinase activation, or RTK. So when you take cell biology, you're supposed to come out of it knowing G protein signaling decently well, and the other big one is RTK. And we have several kinds of RTK proteins, and there are several signals that can activate them, but they use the same early machinery. Kind of like we have a lot of G protein signaling, it uses the same early machinery. Those are the big two. And if you understand generally how those big two G proteins and RTKs work, you can understand a lot of cell biology, because cells tend to use these pathways really frequently. So this starts with an extracellular signal that's a dimer. two of the same protein associated together into a dimer. That's something particular about the signal. And that particular signal is going to bind two identical receptor tyrosine kinases. They're a receptor because they bind the signal. They're kinases because they phosphorylate each other. And they phosphorylate each other on tyrosine amino acids. This is the best kind of cellular name. Receptor tyrosine kinase. Bind a signal, which brings them close together. And when they're close together, they phosphorylate each other. They activate each other. And they end up adding multiple phosphates onto each other's tyrosine residues. And that's what activates them. So I should probably write down here. No, it would be the same as what's up there. I don't need to write new stuff. But they've got to get close together to activate each other and to add activating phosphates onto each other. So we could say step one, the signal binds. Step two, the binding brings the RTKs into proximity. Step three, the RTKs phosphorylate each other on tyrosines. And that activates the whole complex. So it's from here to here that something new happens that I don't have on the words of that slide. So let me see if it's, yeah, here. So once the RTKs are close together, activated, and have phosphorylated each other, this becomes a new shape. This is a new 3D surface. So I'm circling the activated intracellular region of the two RTKs, and this becomes a new 3D surface. It's symmetrical. It's symmetrical because these are identical kinds of proteins. And this complex is symmetrical, and this new 3D surface has binding affinity for a new set of cytoplasmic proteins. And what you see over here are numbered yet anonymous examples of cytoplasmic binding proteins. And see how they're calling it number one, and then there's another number one on this side, because this is a symmetrical complex. And then protein number two, whatever is on this side, there'll be another one on the other side. Protein number three, whatever's on this side is on this side. These you can think of as linkers. That is not their technical name, but what they're doing is they're sensing the new 3D shape that represents active. And they come and bind onto this complex, and they provide a new binding surface for the next set of proteins that are actually going to do something. So these linkers are not enzymes. They're like bridge builders or in-between connectors for the next set of signaling proteins that are actually going to do something. So this red arrow coming down saying activation of downstream signaling pathways. We need other proteins to come to this area, this already seems crowded, that are actually going to carry on the signal. The linkers are boring as far as what they do. I hope no one ever who discovered linkers ever listens to this video. They're boring. And your book is, you know, now we stop numbering them. And your book is calling it adapter. That's linker adapter. Now we come to the proteins that are really doing a lot and are famous, conserved, well-studied, and names of proteins that you should know. And these over here, we start with these are names and processes that you should know. The adapters are boring and we don't have to know their names. But one of the signaling proteins that is recruited to this new 3D shape, and they've taken out the 1 and 2 and the 1, 2, 3 over here just so it's not crowded. They're still there. But one of the proteins that gets recruited to this new shape is called RAS-GEF. Or just, well, yeah. RAS-GEF is recruited to this complex. And there will also be another protein that's nearby, membrane associated, called RAS. RAS. I want to say what RAS-GEF stands for. So RAS-GEF stands for RAS guanylyl exchange factor. Somebody quickly look up what RAS stands for. Google RAS protein stands for. I think Tyler.
Rat sarcoma.
Rat sarcoma. Thank you. The reason that I don't remember what it stands for is it's named after, it was discovered in rats that have cancer, and then its name doesn't tell you what it does. And everybody now who actually studies it always just calls it RAS. And what's important is that you remember that RAS is activated by RTKs and what it does next. So rat sarcoma, yeah. If it's so old, it's like from rats, before they started using mice so much. But that's RAS. And notice that RAS hangs out most of the time associated with GDP. So that's its weighting configuration. GDP-bound RAS is weighting inactive. And what it is waiting for is to get activated by RAS-GEF. So the way that researchers pronounce that is RAS-GEF. And what RAS-GEF does is it helps RAS switch GDP out and bind GTP. It helps with the switch. So RAS normally will be holding GDP as its default state. But an activated RAS-GEF can help RAS get rid of GTP and get rid of GDP and bind GTP. And once it's GTP bound, it becomes activated. Make yourself a note here that RAS is kind of similar to G-alpha subunits. When you look at their sequence and you look at their 3D shape and how they operate, they come from the same ancestral proteins. And they are working in similar ways. So both of them, when they're bound to GTP, that's the active. And both of them, when they're bound to GDP, that's the inactive. Both of them are membrane associated, and they actually have more similar shapes than the book is showing, but they're related in their history. So when RAS is activated, it is able to transmit the signal, but it also, like G alpha, will self-hydrolyze that GTP soon. So eventually that GTP is going to be hydrolyzed to a GDP and a float-away phosphate. But as long as GTP is bound, RAS is active. Okay, this is going to be the last slide for today because the next set of signalers are very important and I don't want to rush through it. So what you should do for next time, for Tuesday, is to make a list for yourself in the same way that we did for G protein signaling. What are the major events up through now to get to the point of having an activated RAS? So you've kind of seen the style that we did that before. We've got extracellular signal binds. It's got to be a dimer. It's binding to 2 identical receptor tyrosine kinase proteins that are stuck in the membrane. It brings them close together. When they're close together, they phosphorylate each other on tyrosines, forming a symmetrical activated complex. Then we have adapter proteins come in onto that new surface. And onto that new surface, that causes activation of RAS-GEF, which helps RAS that's nearby switch a GDP for a GTP, which activates it. And as long as the GTP is bound, RAS stays activated. And what it's going to do is it's going to phosphorylate it is going to activate a protein called MAP kinase. That's where we'll start next time. And that's weird. Religion, no, maybe that's Sydney. Sydney, are you religion 1001?
Yes, I am. I forgot to log out of this one when I logged into Teams this morning because I have to have a separate login for that class.
That's fine. I just, there was one day when Dr. Hudson somehow, I was in Teams and he was sending comments on a faculty team and it kept showing up during my lecture. And it was these comments about this totally unrelated thing about like what we should do in King 1000 next year. I couldn't figure out how to make them stop.
Yeah, I had to like go through registrar to do this because I couldn't accept people into meetings where I was just a student. So I had to get like a faculty Teams login to accept people and to like control the teams. So I just forgot to log out of it and I didn't realize until I was about halfway through this lecture that I was on the religion one.
I figured that was you. I will see you all at 1:45. Any way you attend, I will see you there, 1:45, and just use the same Teams link. Oh, great. I just recorded that whole conversation with